Okay, so this week we're looking at the statement that direct evidence that the induction of dysfunctional spindle checkpoint results in Paxitol resistance has been demonstrated by knocking down BubR1 or BubR1 in MCF7 cancer cells. And the first part of the question is, what does the sentence mean? And it basically means that we have BRCA1, which we know is a transcription factor and has already been seen to upregulate or downregulate proteins like P21 or P51, P53, and we know that BRCA is able to affect these transcriptions and affect the cell cycle through such. What we didn't know is that if in the cell cycle, when you're looking at the metaphase to anaphase checkpoint, if BRCA1 had any sort of hold or control over this. And by examining BubR1, which is responsible for this checkpoint and the bypassing of this checkpoint, by knocking down this protein, we were able to see how this checkpoint was affected in the cell cycle. The best experiment that we could use, or one of the experiments that we could use, is just doing siRNA transfection for BubR1. And what we would expect to see is that in the cell cycle and within the metaphase, we have this metaphase to anaphase checkpoint. And the requirement to get past the checkpoint is the spindle, the mitotic spindle. And we need to have it to be continually stabilizing and destabilizing, and then for all of them to be properly arranged in order for this to get bypassed. And by using BRCA1 or by observing BRCA1 and knocking down BubR1, what we see is that this checkpoint does not get passed. And because it does not get passed, we can see that there's an increase to paclitaxel resistance when BubR1 begins to overregulate due to the spindle checkpoint not being passed correctly. And that's what we can expect to see in the cell cycle.